Laboratory 1, Introduction to the Scientific Method. In any science class, the first lecture will usually incorporate a discussion on something called the scientific method, whether that course is biology, chemistry, astronomy, physics, or geology. Every single science is held by the same standard. This is called the scientific method. And any good hypothesis must undergo a series of rigorous testing, confirmation, and reconfirmation until it can be identified as something called a theory or a scientific fact. So when we talk about looking at reliable information, especially in the modern age, we are always having to check ourselves when we receive new information, i.e., where does this information come from? Does the source have a good reputation? Who are the authorities who believe it? And what are my reasons for deferring to these authorities? So if we hop on the internet here, let's say we are interested in researching the latest reliable science on marine pollution. If we do a Google search, right, we'll get a lot of information, Wikipedia, maybe we'll even get some news articles. But it's important in this class that you understand there is a huge difference between the articles that you would read on a website, such as Washington Post or CNN, and actual scientific research. So if I'm interested in looking at the science and not someone else's interpretation of that published work, I'd come to something called Google Scholar. So here, within this search engine, it will only search scientific papers. So in order for a scientist to have their research published in a scientific journal, they are held to something called the scientific method, and they must prove their hypothesis is correct, and it must be reliable, and the results must be repeatable. So here we can see we have a lot of results here. We can sort that by time. So now we can see this is all since 2020. And so what gets reported in the news, well, sometimes you'll see a paper will be published and it will get picked up by some news station and they'll summarize the work, but oftentimes they get that summary wrong, right? And so it's important that you go to the main source. So there's a couple things to look out for. One, has the work been confirmed? So here we can see cited by 3. That means 3 other scientific papers have used this paper in their study. We have this one cited by 276. So the more citations a scientific paper has, that means the more reliable it is, because that means the work was so useful that it was repeatedly used over and over again. A work that was just published that has no citations yet can be picked up by the media and reported, but it's still preliminary, right? A scientific experiment has to be proven through reconfirmation and retesting many times and through many citations before it's considered a reliable scientific theory or fact. So we have to ask ourselves those questions first. We need to understand, where is this information coming from and why do I defer to it? So the scientific method contains several different variables. And in your first lab, you will be looking at a series of observations. And through each of these, you will list the hypothesis, the independent variable, etc. for your experiment. So let's go through each one of these. A hypothesis, this is a tentative, testable answer to a scientific question based on observations. So that means that all hypotheses start off as something that you observe in the natural world. Maybe you notice that it's been raining for several consecutive days in the winter time in Los Angeles, and you hypothesize that that rainfall is due to an El Nino event. Well, you have to go and test that now. And it's divided into different variables. So in an experiment, you will first make a hypothesis based on your observation. The null hypothesis you will state as well. And this is like the backup hypothesis, because oftentimes in science, we don't confirm our hypothesis. We confirm that our hypothesis was not correct. And so we have to go back to the start and reevaluate. So we have to have a null hypothesis to fall back on. The independent variable, this is a quantity in the experiment that is changed or directly manipulated by the experimenter. So here we can see we have this scientist in the lab, and she is adding a quantity of this chemical into the mix here. So that's the independent variable. She's changing that. The dependent variable is what you measure as a result. So you can see here, she's pouring in this chemical. It's her independent variable. And then the result in these vials here will be the dependent variable. That's the quantity that changes as a result of a change in the independent variable. So she's manipulating the experiment with this and measuring the dependent here. The control variables are very important. The experimental element, which is constant and unchanged throughout the course of the investigation, is known as the control variable. It strongly influences experimental results, and it's held constant during the experiment in order to test the relative relationship between the dependent and independent variables. So what does that mean in a nutshell? The control variables are all of the things in the experiment that you have to hold constant, that you can't change. And this is because, let's say she makes this new discovery and she says, I've cured the common cold, and it's the result of this chemical here, her independent variable. Well, maybe some scientists could come along and say, oh, well, that's pretty cool, but how do you know it was this chemical that cured the common cold? Or she'll say, well, of course it is. But if she didn't keep everything constant, let's say the temperature of her lab varied wildly throughout the day. She didn't have enforced air. So at noon, it was very hot in the lab, and at night, it was very cold in the lab. And the scientists say, well, how do you know your results weren't influenced by temperature changes or influenced by the time of the day that she made the measurements? So she has to hold everything constant because she wants to make sure that what she's manipulating, what she's changing, is in fact causing this result. So everything has to be held constant. Those are the control variables. So let's say she goes through this whole experiment, she confirms her hypothesis, she publishes her work in a scientific journal, and it's picked up in Google Scholar. You can search for it. Does that mean that it's a fact? No, it doesn't mean that's a fact, because that's one study, only one. In order for a hypothesis to become a theory is a well-substantiated scientific fact. And it's the most incorrectly used scientific word in common language and television. You'll hear all the time, even on sci-fi shows, they'll say, hey, Captain, I have a theory. And that's wrong. What they mean to say is, hey, Captain, I have a hypothesis. Because a theory is a fact. A theory is something that has been tested over and over again. It's been confirmed by scientists all over the world in different labs, and everybody, the scientific majority, all agree that that is the best possible explanation for that phenomenon. So some theories that you may have learned about before in high school include the theory of evolution, the Big Bang theory, right? All of those different types of things. It took us decades and major advancements in technology in order for us to substantiate that things like plate tectonics, the Big Bang, evolution, that those are the main mechanisms. And originally, when they started off as hypotheses, and we'll talk about this in the next lecture, right? They weren't accepted. And you may know this about if you've ever learned about Charles Darwin or plate tectonics in the 1960s, but the original hypotheses were criticized and at times even ridiculed because they didn't have scientific foundation. They weren't able to be tested and go through this scientific method. But because we've had such an amazing increase in technology, we are able to now finally test these things directly. And so within the last 20 years, we've been able to identify very precisely a number of important theories that we'll discuss in earth science. So in your first lab, you will be given a series of observations, and you will have to list each of these variables: a hypothesis, an independent variable, a dependent, and a control. So let's do one together. The observation here is that dogs at the local animal shelter, the staff tell you, hey, it's the craziest thing. These dogs seem to really calm down when we play Led Zeppelin over the loudspeakers. So you say, okay, I'm going to test this. I'm a scientist. I must, go through the scientific method. So your hypothesis is that dogs must prefer classic rock, right? They're fond of Led Zeppelin. But just in case your hypothesis turns out to be wrong, you need the null hypothesis to fall back on. So let's say that your results are negative. That would mean the dogs at this shelter, they just have a natural calming mechanism to music. Okay, so now let's look at our independent variables. The independent, remember, that's what you change as the scientist. It's what you can manipulate. And here, that's going to be the musical choice, right? What music you play, that's you changing something. The dependent, remember, that's what you are measuring. So in this case, it will be the volume of the dogs barking, right? Have they really calmed? Let's make sure we measure that. Because, maybe it's just the staff being wishful that the dogs like Led Zeppelin. So we need to quantify that. We always need to quantify something through our measurement. And then finally, our control variables. What are the things that we have to keep constant in the experiment in order to be certain that the musical choice is what is causing the reduction in the volume of the dogs barking? So this is a lot of things: temperature, environment, right? Where are these dogs living? Maybe they're in bad living conditions or good living conditions. What are the dogs' hearing ability? And maybe the dogs can't hear at all. What time of day is the music played? Does that correlate to the time of day the dogs might be fed? Maybe they're responding to having a full belly as opposed to the Led Zeppelin. And the genre of music. So we have to make sure that we hold all those things constant so that we know the dogs are actually calming down because of Led Zeppelin, not something else. Okay, so now we can design some trials. And a real scientist, they'd have more than 4 trials, right? They'd have 100 or a couple hundred. But here we're just going to simplify it. So trial 1, we'll play Led Zeppelin over the loudspeakers and measure the barking. Trial 2, okay, maybe we'll play another classic rock band like Pink Floyd so we can confirm if the dogs like all classic rock or just Led Zeppelin. Trial 3, we'll switch it up. We'll play a completely different musical genre. So maybe we'll play Jay-Z. I hear he's still doing things. So we need to confirm, okay, the dogs like all music. Is it just classic rock or is it just Led Zeppelin? And then probably the most important, which is the trial that you usually should do first, is you do nothing. You need a baseline measurement. You need to know how loud the dogs are barking under normal circumstances because you need something to compare the change with. So in your lab, make sure that when you list all of your variables that you're thinking about potential trials. So if we look at our outcomes, if barking is reduced for the first 2 trials, then our hypothesis is confirmed. Remember, our hypothesis was that dogs prefer classic rock. Let's say barking is reduced for the first 3 trials. That would mean that the null hypothesis is confirmed. The dogs just like all music. And finally, let's say barking is reduced for all the trials. So the dogs are calm no matter what we do. What does that mean? Well, that means that the staff was wrong. The dogs weren't calming down from Led Zeppelin. And so when our hypothesis is shown to be incorrect, that means we have to go back to the very beginning. So what's at the very beginning? The observation. That means we need to reevaluate the observation. And that here was dogs at the local shelter seemed to calm when staff played Led Zeppelin over the speakers. So that has turned out to not be true. So we'd go back to our observation and we'd make a new hypothesis. Maybe the hypothesis is the staff at this dog shelter are under a lot of stress and they imagine that the dogs are quieter when they play Led Zeppelin. Maybe the barking is just harder to hear when they're playing Led Zeppelin. In any case, if your hypothesis is not true, you have to go back to the beginning and reevaluate your observations to form a new hypothesis. So finally, I will leave off with a helpful cartoon that can give you an idea of how to sort through the enormous amount of scientific information that is out there. Especially you guys, as most of you are taking this as a general science requirement. As you go throughout your life, you're going to be, burdened with many new scientific hypotheses that the media picks up. And you need to do your research before you peddle something as fact. Remember, theories take decades to confirm. So keep an eye out. There's two different kinds of scientists, right? The good and skeptical scientist and the bad, unskeptical scientist. So a good scientist holds observations above theory. They don't break rules of logic. They're nice in that they'll answer questions. They're helpful, polite. They will give out all their data, their methods, everything that other people would need to repeat their experiment, because they're not interested in ego. They just want truth, right? They want to find the right answers. So that involves making all their information available, right? They're polite. They even explain what would falsify their hypothesis, right? They present the counter argument. On the other hand, you will see this all the time. Maybe a new scientific article is published and CNN picks it up and you're reading it and it sounds really dubious. And so you go and look for the original scientific article that they're reporting on. Let's say you read the first paragraph called the abstract, and the scientist seems to hide their data. They're very secretive about it. They won't debate or answer questions. Maybe they're a bully. They'll name call. They'll threaten. Oftentimes they have sort of a blind faith in certain systems, right? They'll say, oh, well, it's from Caltech. It can't be wrong, right? They don't even look to read the data, right? They sort of have this blind faith. They use circular reasoning. So be careful for that. A real good scientist will be happy to give you all their data. They're helpful. So when you're going through, pay attention for those different types of things.